Stepping in for this session. We've got a bit of a mixed session. We've got a session, first part of it is going to be on birds and avian communities, and then we're going to move into some vegetation dynamics. Just quickly before we get going, if your name is or you are related to have somebody that you know is Mrs. C.B. Marais, we have your Standard Bank card here, and if you don't come and collect it, we're going to try and figure out the PIN and see if we can get some money for tonight. All right, so with that, I think we are ready to go, and our first talk this afternoon is by Craig, and Craig's going to be talking about auditory cues and vultures and other scavengers. Thanks, Craig.
Thanks, Dan. So yesterday there were quite a few talks that really illustrated the impressive diversity of herbivores in the majority of African savanna ecosystems. They occur in all shapes and sizes, and in most places there's numerous species occurring sympatrically. Now this diversity and abundance also means that there's a lot of biomass available to carnivores, and it's probably something that's been quite important in the evolution of perhaps an equally impressive array of large carnivores in these ecosystems. Now, these large carnivores are obviously dependent on capturing their prey, and in doing so, they also create other or secondary foraging opportunities for scavenging species. Now, looking away from the mammalian scavengers, there's also numerous bird species whose diets include some component of carrion, and this is perhaps best epitomized by the vultures, who are obligate scavengers. Now, when it comes to foraging strategies have formed an important part of animal ecology, and that's because of the direct link with foraging success and an individual's fitness. And when it comes to the vultures in particular, and dependence on carrion is not very predictable in its distribution, and it's widely distributed over large areas, and it's not necessarily so easy to find. So unlike other vulture species in other parts of the world, or some of them at least, who are able to use olfactory cues, the vultures in Africa are thought to only rely on vision. And that can obviously be to locate the carcasses or the species that have already arrived there and feeding on them. So these could act as cues to lead them into their foraging opportunities. But if we look a little bit closer at this picture here, you also see there's a very prominent ear. So what about sound? Could that act as a cue, a reliable cue that could lead them into foraging? Well, if you think of it, the whole process of predation often results in a lot of noise. Killing large prey takes quite a while, during which time there's often distress calls. But in particular, once the animals are dead, then it can be several hours with very noisy intra and interspecific interactions, which then could signal possibilities of food. And these recordings of these same interactions are readily used by carnivore biologists to survey large carnivore populations, in particular lions and spotted hyenas. So you broadcast these sounds through a powerful sound system, and depending on the terrain and ecosystem, these sounds often travel many kilometers, and in doing so, you attract the carnivores to the calling station, and you can survey and census their populations. Now, because these are typically focused on lions and spotted hyenas, they're done at night. We did calling stations in the Serengeti ecosystem, but we did them during the early morning. And this then gave us the opportunity to also look at avian scavengers that potentially could be attracted, which otherwise wouldn't be possible if this was done at night. Based on our current understanding of these species' foraging ecology, we hypothesized that the response to any calling stations would be evoked only by visual cues, not the use of sound at all. And based on that, then, we predicted that the arrival of avian scavengers at calling stations should be preceded and facilitated by these other mammalian carnivores or scavengers. And on top of that the arrival of these birds would be positively correlated to the number of mammalian scavengers or carnivores already there. So the more hyenas or jackals that are present, the greater the probability that these birds could see them and then thus be drawn in. So if we look at the results, we had approximately 300 calling stations or calling sessions through a prolonged period. Birds were attracted to 122 of these, so approximately 40%. And in 27 of these instances, the birds arrived first before any other mammalian scavengers or carnivores. So that, given that there was no carcasses, there was no bait, the only cue was sound, gives pretty conclusive evidence that they do in fact or are able to use sound in the foraging. We attracted in total almost 500 birds. 320 were vulture species, 5 different vulture species. The only species that didn't arrive first before any other birds or any other mammals was Ruppel's griffin vulture. All the others did. And then as for the second prediction, we expected a positive relationship between the number of carnivores already present and the arrival of the avian scavengers, but this was in fact the total opposite. So the majority of birds arrived in the absence or near absence of mammalian carnivores. So just summing up, these results show us that the birds do in fact or are able to utilize sound, something that maybe hasn't received much focus before. And this whole experimental setup was not targeting these birds or addressing this question in particular, so the data was rather fortuitous. So I think that in future it would also be possible to set up better experiments that are better able to dig into the finer details of how this might work and also the differences between the different species. This work is published if you're interested, that which I presented today, as well as some other analyses that really looked at the potential facilitatory role of the different species that were attracted in. So you're welcome to have a read there. Thank you.
Thanks very much, Craig. We are out of time, so if you've got a question for Craig, please grab him afterwards or this evening. Next up is Tom. We're going to stick with vultures, and he's going to be telling us about vulture nest sites or surveying them using LiDAR. Thanks, Tom.
I'm Tom Lautenbach, and I'm going to be doing some stuff or telling you about my master's work. And I'm looking at nesting tree characteristics of white-backed vultures in Kurungani Game Reserve. So what got me interested in this is I've been more focused on wanting to figure out, like, why do these vultures choose the trees that they choose? What variables or attributes are they choosing for? Traditional methods, obviously, for measuring trees it's very time consuming. It's somewhat limiting with what you can measure, especially if you're doing tree height or tree canopy width. If you're using a small golf rangefinder, it's one laser, the accuracy of that is somewhat suspect. But with LiDAR, our LiDAR in particular, which is also a form of measuring things with a laser, but we have 750,000 lasers coming out every second, which gives us a really high detail 3D model of a tree. And also we're matching up our data with ground-based GPS stations and global towers like TrigNet, so the accuracy of our measuring is very high. Well, it is accurate. And from that, I'm hoping to be able to take this kind of, the data that I pull, put it into some sort of predictive model where we can, for future LiDAR data, take that, put it into the model, and the model will say, okay, cool, this area looks very suitable for white-backed vulture nesting because there's optimal trees here for them to nest in. So went to Kurungani Game Reserve, which is just to the east of the Lubombo Mountains there. And we flew the HALO, which is the Harvard Animal Landscape Observatory, which is a drone-based combination of LiDAR, thermal, and RGB. We mapped roughly 35,000 hectares of subsamples, and then took known GPS points of vulture nests, overlaid that onto CHMs, DTMs, and orthos. And from that, I was able to go down to and extract measurements of different variables at an individual tree level. I did that for the known nesting trees, and then I also did that with a number of random trees without any nests in them, ran them against one another, and we'll do some more of that in some RSFs or resource selection functions, and hopefully that will be able to give me more insight to what attributes and variables that these vultures are choosing for within the nest. Why are these vultures choosing the trees that they choose? So some of the measurements that we are able to measure reliably are tree height, tree canopy width, This one's really cool. We can also look at canopy ruggedness. We can also then look at tree canopy area and the nest location within a canopy, which I thought was quite cool too. If our LiDAR is strong enough and can penetrate through into the vegetation deep enough, we can also get, if we can run that data through something called a quantitative structural model, which is a QSM, and it can give us first, second, and third order branch level, which gives us a better picture of like the structure of the tree. So then with some preliminary data, we're seeing here that they do like tall trees. They like big trees. None of them were in any trees below 10 meters. And I think this is quite important in context with we're losing all of these tall trees in the savanna ecosystems due to multiple different reasons. So it's quite an important thing for conservation of vultures to, take note of this. Other stuff, canopy area is also a big one too. They really like large canopies. And with the canopy roughness was something that I wasn't expecting and I thought was really cool, is they are selecting with trees with more variation in the canopy height within the canopy, like the roughness. They don't want these flat crowns, which one would have thought. But yeah, so that's just some of my preliminary data and what I've been up to. Thanks.
Thanks, Tom. Just stay there. We've got about a minute for questions. So if you've got a question for Tom or for Craig, there is an opportunity. Who have we got there? Sharon.
Hi, Tom. Very interesting. Have you tried to do this LiDAR work for other vultures besides the whitebacks? Is your LiDAR able to pick up things like hooded vulture nests in these
No,
big
so
trees?
actually we aren't able to see the nest itself in the LiDAR, so that's why I'm having to take the GPS points, and then we just confirm that with the RGB that we lay on top.
But then you would be able to still use the LiDAR to map the characteristics of that particular tree if it was
Yes, for
like
sure.
a hooded or...
Yes, yeah, we'll
Okay,
be able to do that.
cool. I'd like to chat to you a bit afterwards.
sure.
Thanks.
Excellent. Thank you very much, Tom.
Cool.
Right. We're still sticking with vultures, but we are going to have a 15-minute talk now by Tawanda, and looking forward to hearing what you have to tell us. Thanks, Tawanda.
Good afternoon. So I'm Tawanda Tarakini, and I'm from Chino University of Technology. I'm going to be presenting our short work from Wange National Park, north of where we are right now. The system almost similar, but quite different in terms of water availability. So I've got my colleagues that I worked with for this project. So it's mostly looking at the interaction of vultures on carcasses. So thanks to my first two presenters of this session, they've already done the groundwork for me. So I'm just going to go straight into the questions that I had. So we wanted to find if vulture abundance and composition vary across habitat types and also the state of the decomposition of the carcasses. Secondly, we wanted to look at the specific interactions of the vultures in relation to when they are tagging for the meat, who is winning and who is losing, and probably discuss some issues around that. So as I've already highlighted, the study we conducted was in Wange, and we categorized the carcasses that we found, whether they were fresh, actively decaying, late decay, or the carcass was almost in a drying state. We used the camera trap approach, where we, after discovering the carcass, we would set up the camera about 50 meters from the carcass, and the camera traps were set to record 2-minute videos after every 10 minutes. So that was during all the daytime hours when there was a light to ensure that, and also saving the batteries. So of course we used the time-lapse software to sort out our data, and we created 2 variables, who was on the offensive side when the vultures were fighting for the meat. So if the one which was chasing the other one, we will call it the offender, and the one which is being chased or which is being having meat grabbed from them was the victim. So of course we created several data sets from that for each species where it was the offender. And from the view of the camera traps, we were also able to estimate the abundance of the vouchers by just counting. And I have to say at this point, not all the interactions which were recorded on the camera traps were visible in terms of identification of the vouchers, because sometimes the interaction will be right in between the group. So just going straight into the results, during our small time that we had in 2021, we monitored 6 carcasses belonging to the elephant, giraffe, and kudu. And some of these carcasses were monitored over a long period from the fresh state until they were dry. We had 4 voucher species that we recorded on the camera traps: the white-backed, the lappet-faced, the white-headed, and the hooded. And of course, we had other non-voucher species, such as the yellow-billed kite, the marable stork, the pied crows, and of course, the hyenas, which were captured on the cameras. Just to have a look on the proportions that were there, we almost see all the species being represented, except the case for the a fresh giraffe carcass, whereby almost 90% of the vouchers that we found there were from the white-backed. Moving on to issues of the abundances across the habitats and the species, we found that there were high voucher abundances on fresh carcasses, but in the open woodlands, although we had expected greater abundance in open areas, just like the first presenter was highlighting that if the African vouchers are using the eyesight for cues, we would expect them to discover carcasses earlier. We found the opposite, in that they were actually more in the closed habitats. It is only the lappet-faced vouchers which were found in relatively higher numbers in open woodlands. And of course, when we look at it, the competition that can be experienced even in those closed woodlands, we might begin to have a discussion on the level of disadvantages of a species when it is in an open or closed woodland. And this might even be affected by the current vegetation changes due to herbivore activities. So moving on to cases whereby the white-backed vouchers were in the offensive mode. These were very notorious because they seemed not to respect anyone. They were offending everyone. But the attacks on white-headed vouchers were less, except in the cases where the carcasses were very fresh. So could this just be an issue of the numerical advantage of the white-backed? We also realized that when the carcass was getting dry, there was very little of tossing around for the meat. Moving on to the wooded vouchers, they seem not to offend anyone except the intraspecific interactions. We know that on the gathering that they do on around carcasses, they are usually few in numbers, and also their size inferiority might influence that. But literature says that they also feed on pieces of meat and bones that may fall to the ground, and they also do their own hunting. So that could be used as a discussion whether they really depend on these carcasses or not. Moving on to the white-headed voucher, we realized that they were dominating over the lappet-faced and other non-voucher species. So we take note that the white-headed are also quite large in size, and we expected more respect, but they are usually less numerous on the carcasses themselves, And they are known to to use other sources of carrion. So for example, catching fish or mungus. When we looked at the cases where the lappet-faced were winning in their interactions, we also realized that they were only recorded in intraspecific interactions. So we also asked ourselves whether this could be a numerical disadvantage for them, although the literature says they are quite dominant over most voucher species when they are on carcasses. But we also know their critical role in that they've got the muscles and the beak to tear up carcasses. So that could also partially explain on the fresh carcass when they were found in relatively higher numbers. So just trying to conclude, of course, we had very few carcasses to write our story, but we believe that we can say a few things. So we had relatively medium to large carcasses, starting from kudu upwards. So we missed what could be happening on smaller carcasses, like the dikers and smaller than that. But what we realize is that if you put aside the case of the fresh giraffe carcass that we found, there were more or less similar numbers and compositions across the carcass, which kind of suggests that the meat here was probable the determining factor on the composition. But when you look at the cases for the lappet-faced and hooded voucher, and we look into the future, the projections, especially with climate change, drying up, if we have got less meat in the system, this kind of pressure might push them into further disadvantageous positions ecologically. And so as managers, I think we need to be looking into the future and try to see. Of course, at the moment, we do not seem to have any reason to worry, but the future projections might make us to think about those. If we look at the current efforts that we are making as managers, especially in the Zimbabwean context, we seem to be focusing on other threats to vouchers, for example, poisoning, illegal trade, and human perceptions against the vouchers. And very little is being focused on what amount of effect does food availability have on these vouchers. Lastly, I would like to thank the Zimbabwe Parks and Wildlife Management Authority for allowing the first author to do the work in Sinamatela, and also Bejani Trust for providing the camera traps. Thank you.
Tawanda. There is some time for questions. We've actually got 5 minutes, so if you've got any questions for Tawanda, and in fact any of the previous two talks, you can ask them now. Or not. Right, I wonder to see if Craig's still around. Oh, we've got, okay, right, there we go.
Are there differences in the offensive behaviors of the different species? Is it like open-wing threats, attacking with bigs? Are they all offensive in the same way, or do they use different behaviors?
They do use slightly different behaviors. Of course, it's something that I did not present here, but you see more of the white-backed do of the probing, chasing around, and if the white-headed had to interact, for example, one of the things that I would remember, it's actually grabbing meat from the mouth of another eating voucher. And if you look at the competition that could happen between the white-backed vouchers and the marable stock, it would be actually a tug of war, maybe pulling the intestines or something like that with 5 vouchers and the marable stock on one side. So it was quite interesting drama also.
Tawanda, thank you. Any idea about the role of livestock carcasses in the area? There must be livestock carcasses inside the protected areas, predated by lion, and maybe outside the protected area. Do you have any ideas or future research on that?
Sorry, I missed your word. Any idea on the?
Livestock
Livestock.
carcasses.
Okay, indeed, the system that we are working on, it has got some communal areas and farms around it. So there are sometimes livestock carcasses which can be discovered around, and sometimes inside the protected area, especially when you look at the eastern side. And these also present themselves as available feed. Unfortunately, we were not able to document such in our study, but our general knowledge of the area there is. Actually, to feed on to the earlier presentation in the last session before lunch, this should also feed on to the general discussions that we're having about disease transfers between livestock and wild animals and the One Health concept.
Thank you very much, Tawanda. Thank you. Right, folks, we're going to have one more bird presentation. It's going to be a 5-minute presentation before moving on to our vegetation section for this afternoon. And we've got Sharon up to give us that 5-minute presentation talking about pied crows. Thanks, Sharon.
Okay, thanks very much, Dan, and good afternoon, everybody. So today I'm going to be doing a brief discussion on basically the rising concern and the knowledge gaps that are surrounding pied crows, their distribution and their abundance. So pied crows are indigenous to Southern Africa, as many of you know. They are distinct with very bold black and white markings, and they are widespread throughout South Africa and common in many regions of the country. But in the last decade, the Southern African Bird Atlas Project, and for those of you that don't know what that is, that's a citizen science project that is collecting large amounts of reporting data of sightings of birds in the country, in fact, actually even nationally. But there has been an increase in reporting rates of pied crows, particularly in these southwestern areas of the country. So if we just look at this map, all of these blue pentads in this area, ranging from this kind of lighter shade of blue right up to this very dark, deep shade, that's basically showing you an increase in reporting rates of between the lighter shade is around about 5 to 10%, and then these darker, deep shades of blue are between 50 to 100% increase in reporting rates of pied crows. So some of these regions in the southwestern area have experienced between 25 to 50% more sightings in the last 10 years. There's also been an increase in reporting rates in this northeastern area of South Africa, but this is a lot more variable, patchy. But in general, this increase that we're seeing is actually reflecting a global trend, with many countries worldwide reporting more sightings of members of the corvid family in general. So what does this mean for Kruger? So if we zoom into Kruger, again, the blue pentads are basically an increase in reporting rates, and we can see that this varies in intensity, but it is mostly along that western boundary of the park. And there are two slightly lighter pentads that are showing an increase in reporting rates within the park, and that's actually the Mopani camp and the Letaba rest camp. There's also been an increase in ad hoc sightings of pied crows in Skukuza, and there is a rising concern about this amongst a lot of people that are seeing these birds inside the park in Skukuza. But what is surprising is that there are also declines in the reporting rates, and these are actually indicated here by these red pentads. And these declines are happening inside and outside of the park. So
is there cause for concern, and should we be taking immediate action? But to answer this, we really need to assess the status quo of pied crows within Kruger. So we know broadly that there are three factors that are driving this increase and expansion of pied crows into these new areas. And this is mainly climate change. It appears that these birds are moving into areas of warmer temperatures. Of course, the expanding linear infrastructure, which is like cell phone towers and electrical infrastructures, and that's providing artificial nesting opportunities for these birds. And there's, of course, increased food availability from frequent access to human waste and even to roadkills. But what we don't know is the impact of this increase and expansion into these new areas and on the local biodiversity. There's negative impacts on the biodiversity are only evident at this stage in some cases. Most of the research has showed a negative impact on tortoises, especially in the Karoo, and a couple of incidents of vultures as well. But the data confirming negative impacts on other indigenous species is actually lacking, especially quantitative data. And also the ecological role that these birds are going to be playing in these newly inhabited ecosystems is completely unknown. So clearly there are some very important research gaps that need to be investigated. We need to determine the status of the pied crows in Kruger and quantify perceived negative impacts of crows by looking at their breeding biology and nesting ecology, and to monitor interspecific behavior, including competition and predation. And of course, we need to determine and control local anthropogenic drivers of the crow population growth, perhaps instead of the crows themselves. So the case of the pied crows really isn't as clear-cut as one would have thought, and there's clearly a bit of lack of scientific evidence, and acting on impulsive conclusions could be far more detrimental than actually the pied crows themselves. Thank you.
Thanks very much, Sharon. We are unfortunately out of time, but if you've got a question for Sharon, you can grab her afterwards. Folks, we're going to move into the kind of second half of the session, which is about vegetation dynamics now. But just before we do, and I introduce our first speaker, I'd just like to remind you that there are two posters linked to the bird part of the session, one by Chifiwa Nangambi and one by Don Tai, out in the foyer, so please go and have a look at those. But to kick off our vegetation dynamics section, we've got Max, and Max is going to be telling us about pathways of resilience in savannas. Thank you very much, Max.
Thank you very much. My name is Max Rietkerk. Before I start my presentation, I would like to first set the scene with a small video, which will take about 5 minutes, and after that I will start my presentation. There we go.
Some organisms are really clever. Plants, for example, improve their own growing conditions by increasing water infiltration. In this way, they can survive in environments that are actually way too dry. But it also makes them prone to suddenly disappear. In the face of alarming global changes, there has been much talk about such tipping points in Earth system elements. A tipping point can exist when the same environmental conditions can support two different possible ecosystem states, such as a vegetated and a barren state. A stable state is like a ball in a valley. If conditions become drier, this can cause the ecosystem to tip to the bare state. Water input then needs to improve a lot more for the vegetation to tip back into the system. A stable state means that when the vegetation is disturbed, there is a bit less, But this is only temporary. When a disturbance is too large and the system ends up below this dotted line, then it tips to a bare state. Plants can return when they do so in large enough numbers. Tipping is not so convenient for us. Healthy ecosystems are important for people, so we like them to be resilient and not prone to sudden large changes. Luckily, the notion of ecosystem tipping depends a lot on the spatial scale that you're looking at. Some organisms, such as plants in dry environments, can form spatial patterns. As the environmental conditions change, the patterns adjust accordingly. Through these pattern adjustments, the vegetation can adjust freely and gradually through what we call a booster balloon, avoiding the tipping point. Organisms, especially trees, do take some time to grow back, so if the climate changes too fast, the system can still tip. These insights are obtained mostly from mathematical, physical and ecological theory, but models are clear, simplified versions of the real world, whereas the world is actually more like a big mess. Such spatial heterogeneity can also cause spatial pattern formation, for example at the border between savannas and tropical forests. You see, plants can be rather selfish. They worsen conditions for plants of another kind. Tropical forest trees block sunlight so that savanna vegetation cannot grow. And savanna vegetation stimulates fire. Savanna trees are adapted to fire, but tropical trees are not. So savanna plants are bad for forest plants, while forest plants are bad for savanna plants. This results in two alternative stable states. Both savanna vegetation or tropical forest can exist for the same environmental conditions. In a spatially heterogeneous environment, a local disturbance in a patch of tropical forest can lead to stable spatial patterns. These patterns can also adjust to climate change and create stable coexistence states that allow the ecosystem to avoid the tipping point. So, in a world with underlying variety, after a disturbance, the system isn't immediately pushed to either the savanna or the tropical forest state, but it ends up in a coexistence state. The underlying environmental heterogeneity is necessary to get this coexistence of states in one spatial domain. We still must be cautionary with changing our Earth system because tipping can still occur under certain conditions. So it is really important to consider spatial processes when studying tipping points. Research on resilience in a spatial context also shows what happens when we take away space. With limited space for natural processes, ecosystems can still tip. So, if we implement policies that allow space for natural processes, we create landscapes that are actually resilient in the face of climate change.
Thank you very much for watching. This was the easy part for me. Now starts the hard part. So I will be talking about spatial resilience in complex savanna systems. So first we had the video. I will talk a little bit about the booster balloon that was just introduced, and then a little bit about coexistence states, and finalizing with the conclusions. And here you see the literature that I based my talk on. So earlier we thought that these self-organized patterns that were introduced also in the movie were actually indicative for a tipping point. However, more recent model analysis actually revealed a whole different story. And that story is the occurrence of what we call a Busse balloon. What you see here on the slide is Mr. Busse, actually a person from hydrodynamics studying convection cells, and he invented the Busse balloon. And actually the Busse balloon can also be found in the models that we use for dry savannas, as indicated in this figure. This is because the patterns that appear in these models, they appear much more stable, or sometimes we call them sticky, than earlier thought, and they evade the tipping that was predicted earlier. And within the Busse balloon, many stable patterns can exist with different wave numbers. So these patterns look more or less like this. Those are what we call Turing patterns in dry savannas, this is. Differing in amounts of rainfall and also differing a little bit in topography. For example, the most right picture is a shallow slope in which you get striped patterns, what we call tiger bush, and the stripes, they walk slowly upslope the hill. So those are dynamic patterns, and the rest is stable patterns. And we understand them quite well, because with simple models, we can actually generate all the patterns that we see, and they make all ecological sense. So we think we understand this quite well. So what we have here is a Busse balloon, which means pattern adaptation and smooth transitions instead of tipping transitions. As a consequence, we have a narrow hysteresis loop and smooth recovery, as opposed to what we thought earlier. And the things I want to bring more attention to now is, as you can see in the Busse balloon, oops, go back. You can have changing environmental conditions while the pattern remains similar, and at the same time you can have the same conditions and have a whole range of different patterns, which sounds counterintuitive, but it is not, as I can show. So we gathered a team of experts to actually try to validate these two specific predictions from the Busse balloon. And we took two areas in Somalia, which were quite extensive in their vegetation patterning, and especially in these areas, those are tiger bushes, so the striped patterns on shallow slopes that move slowly upward. So what you see here is a picture of what we think is a real Busse balloon. Why is this a real Busse balloon? This is because you have one side with what we think equal environmental conditions, but you see a whole different lot of patterns with different wave numbers given that similar condition. So we think that this is a picture of a Busse balloon, and we are proud to say that Mr. Busse agrees with that. So what we can do is we can actually validate simple models in different ways, and this is what we did here. So the first prediction was that given a certain environmental condition, here we chose slope on the x-axis instead of rainfall, because remember we had one area with the same environmental conditions. So the changing thing here is slope, and you see the model predicts a whole range of stable patterns, a whole range of wave numbers. And actually we find that also in the data. Given a certain ground slope, we find a whole range of stable patterns with different wave numbers, given the same environmental conditions, which makes it until now impossible to predict if you have a certain environmental condition, what the wave number of your pattern is. A little bit more in-depth validation. The model predicts that with increasing biomass, the model predicts that with increasing wave number, both the biomass increase and the migration speed of your patterns moving upslope decreases. And this is another level of validation of this very simple model. So what we had is a Busse balloon, but of course there is a whole range of parameter space that we did not explore yet, all this space. And what we think is that there is a whole lot of more patterning possible within this parameter space, and that is what we call coexistence patterns. So what you see here is all kinds of spatial patterns between the two stable states of the simpler model, but then they occur in specific spatial configurations. This is a savanna system bordering a tropical forest, and this is a shrub invading tundra ecosystem. And these coexistence states can easily occur because of very general and omnipresent mechanisms. That could be local disturbance, that could be spatial heterogeneity, it could be noise, it could be boundary dynamics, it could be phase separation, it could be. So then a different picture arises as compared to earlier, in which, for example, multiple stable states and tipping points are predicted between tropical forests and savannas, between savanna woodlands and savannas, and between savannas and deserts. We think that those multiple stable states and tipping points can be much constrained because of spatial patterns that are apparent along this whole gradient, as you can see here from aerial pictures. So very interesting aspects of the things that we are exploring right now is what we call boundary dynamics, because those boundary dynamics can actually lead to reverse tipping or reverse transitions as a consequence of spontaneous self-organization. And we think that we can find those patterns also in the real world. And more importantly, those boundary dynamics probably could lead to novel focal points for human intervention to manage those systems to get it in the state that we want. So to read most of the theory about this is this paper. And I would like to draw your attention to a very nice project that we have at the moment. We started a year ago. That is an ERC resilience project on spatial resilience of savanna systems and tundra systems, which we would like to compare. And we are developing new theory along with new data acquisition and model validation. And we have quite some vacancies open at this moment. And there is a poster in the lobby, and you can just scan the link and see the vacancies and apply for the positions. So concluding, the concept of tipping is too simplistic for spatial systems. All systems are spatial. So we need new concepts, such as spatial pattern formation. And spatial pattern formation leads to multi-stability and large-scale resilience. Thank you for listening.
Thank you very much, Max. We have got a little bit of time for one burning question, if there is one in the audience. Doesn't see Craig. Where's my Garmin? I need to get my steps in.
Thank you. So do these same concepts apply to pattern formation in development, like zebras and leopard spots and all that sort of thing? And how would you compare and contrast between the two?
Yeah, thanks for the question. Indeed, the theory of pattern formation is very general and started out with a chemical reaction. Actually, it started out with an equation of Alan Turing. And this theory is wide applicable to all kinds of systems and apparently also to ecosystems. There must be differences and similarities, of course. Where those lie, we still have to explore. But the similarities are really striking. Actually, the first model on this tiger bush patterns is one-to-one equivalent to a chemical model, which we call Gray Scott, but also wasn't aware, I think. But it was just chemistry, what he wrote about, thinking it was ecology. Yeah.
Thank you very much, Max. Next up is Brad. Brad is going to be telling us about the physiological mechanisms of tree encroachment. Thanks, Brad.
Thank you very much. For the last few years, we've been looking at the role of elevated CO2 in driving bush encroachment, and we've been particularly interested in how CO2 interacts with drought, disturbance, and competition. But we're also acutely aware that there are a select group of trees that are involved with encroachment, and they have very closely related trees that are not encroaching. And very little is known why some trees are encroaching and others aren't. So a big collaboration with lots of people and funding from NERC and the Department of Science and Innovation, we designed a big experiment at the Rhodes University Elevated CO2 facility where we grew 12 acacia seedlings, 6 that were encroaching and 6 that were non-encroaching, and these were grown either with grass competition or without, watered or under natural Grahamstown rainfall, and at 400 or 500 ppm CO2. And we measured all sorts of things, as I'll show you in a minute. It was a really massive experiment. This is the harvested pots getting near the end of the experiment. It was something like 600 odd pots, each weighing 50 kilos, 14 tons of soil. It's a big thing. These are the species that were in our experiment. And the thing to notice is that the encroachers are all Vachillias, whereas the non-encroachers included Vachillias and Senegalias, and this adds some complexity to our analyses that I'll talk about just now. These are the actual species replications. And this is just to show the kind of scale of the experiment. And we had lots of people involved in the final harvest, so we could measure lots and lots of different responses. So biomass, allocation, gas exchange, chlorophyll fluorescence, plant hydraulics, allocation and branching, nutrients, stable isotopic compositions, all sorts of things. I vowed never to get involved with a four-way interaction experiment, but here we are with one. And you get these massive outputs which are almost incomprehensible. And so in order to deal with that, we had two approaches. We used mixed effects models with species as a random factor to see if the treatments were being significant and whether there were differences between encroachers and non-encroachers. But the problem is with our particular species bias, you can't really tell whether it's differences between Senegalia and Vachellia that are accounting for those differences. Excuse me. So we also did some phylogenetically corrected stats, these MAC-GLIMs, which use the phylogeny to weight the statistics, and that can really tell you whether the differences are because of the encroacher, non-encroacher groupings, or whether the treatment effects are independent of phylogeny. So I'll just talk you through one response variable. This is total biomass. There was a significant encroachment by competition interaction, which means that competition reduces the biomass of the trees, and this effect was bigger in the non-encroachers than the encroachers. The size of the images scales with the response. For total biomass, we also saw a CO2 by water interaction. So when the plants were watered, elevated CO2 increased their biomass in both the encroaching and non-encroaching species. But it was only significant with the LMER, so this may well be Senegalia, Vachellia differences that we're looking at. Also for total biomass, there was a weak overall biomass of encroachers was bigger than non-encroachers. So we could go through this process for all those response variables, and I could keep you here for the rest of the afternoon, and I know you would be entirely enthralled. Or I could try and do these flow diagrams to summarize some of this data. So this is total plant biomass, and you can see the elevated CO2 by water response. The encroachers were bigger than the non-encroachers, and that grass competition decreased plant biomass, and it did it more so in the non-encroachers than the encroachers. Now, plant biomass is made up of above ground biomass, and here we see again the CO2 plus water effect increasing above ground biomass. Again, grass competition decreased above ground biomass, and more so in the non-encroachers than the encroachers. And there was a weak, marginally significant effect of drought, which was bigger in the encroachers than the non-encroachers. Sure, could I get some water there? Of course, total biomass also has a root component, and this is coarse roots. And again, we saw that grass competition decreased root biomass, and the effect was bigger in the non-encroachers than the encroachers. And competition also was different between the watered and the drought plants. Super, thank you. Just take the little sheet. Thank you. Okay, so we can also divide above ground biomass into leaf biomass. And again, we see the effects of grass competition, bigger on encroachers than non-encroachers, and encroachers more affected by drought. Stem biomass was only affected by CO2 and watering, whereas spine biomass was affected by all sorts of things. There were bigger spine biomass in encroachers than non-encroachers, and again, grass competition decreased the spine biomass more in non-encroachers than encroachers, and elevated CO2 increased spine biomass in both groups, and drought decreased encroacher spine biomass more than non-encroachers. So you can see that there's a pattern emerging here. And we can also look at allocation. So we saw in the previous slides that it looked like the encroachers were more resilient to grass competition, and it seems like this might have something to do with root to shoot ratio. So with grass competition, the encroachers increased root to shoot ratio more than the non-encroachers, and this was independent of phylogeny. So if they're making bigger roots, the carbon has to come from somewhere, and one of the places that it's coming from is the leaf mass fraction, and the leaf mass fraction decreased more in the encroachers than the non-encroachers, and there's also some CO2 effects on leaf mass fraction. We also saw that carbon came from spine mass fraction, with a bigger decrease in spine mass fraction in encroachers than non-encroachers. And lastly, stem mass fraction responded to drought, and it did so more so in the encroachers than the non-encroachers. We could also look at some physiological traits, and this is photosynthetic rate. And very interestingly, with grass competition, elevated CO2 increased encroacher photosynthetic rate more than non-encroachers. And there were also effects on stomatal conductance, where in the watered plants, elevated CO2 decreased the stomatal conductance in encroachers much more than non-encroachers, and this would have big water use efficiency implications. On a subset of plants, we also measured hydraulic conductance, and that's how easily water can move through the xylem and the stems, and we found that encroachers had 26% higher hydraulic conductance than non-encroachers, and we measured the hydraulic vulnerability, and that's how easily xylem can embolize, and we found that encroachers were 12% more vulnerable than non-encroachers, and in both groups, elevated CO2 increased the vulnerability of the hydraulic system. And yesterday, Edith spoke about photosynthetic acclimation in these groups, and she showed that the encroachers upregulate two of the important photosynthetic parameters, whereas the non-encroachers downregulate those parameters. And one of the ways that plants could upregulate photosynthesis is by having a better source of nitrogen, so that they could build more rubisco. And what we found was that the encroachers had 65% more nodule biomass than the non-encroachers, and that nodule biomass responds very strongly to both watering and elevated CO2. And we also determined the nitrogen stable isotopes, and we showed that the isotopic ratio was 33% lower in encroachers than non-encroachers, and this implies that the value is being pulled down towards the value of atmospheric nitrogen, implying that they're acquiring more nitrogen from the atmosphere via their root nodules. I won't worry about this at the moment. And so in summary then, it seems like when we compare encroachers with non-encroachers, the encroachers are bigger, they've got bigger root systems and more spines, they're less affected by grass competition, and they're more responsive to both groups are responsive to elevated CO2 when watered, but the encroachers are more sensitive to drought. The encroachers are better grass competitors, and they increase their rooting under grass competition at the cost of allocation to spine and leaves. They increase their photosynthetic rates with elevated CO2 when faced with competition. They decrease their stomatal conductance more with elevated CO2, giving them a greater water use efficiency. They're hydraulically more conductive, which means they could possibly get more water to their photosynthetic surfaces, but they're more vulnerable to embolism. They also upregulate photosynthesis, in our case when the plants were watered, and this might be because they've got more root nodules and they're requiring more atmospheric nitrogen via those root nodules. So we think that across this collection of traits, we now have quite a good explanation of why some species are encroaching and others aren't, and also we have some idea of how elevated CO2 might affect encroachers versus non-encroachers, and we certainly think we've got enough data now to do a more nuanced model where instead of modeling C4 grasses against C3 tree seedlings, we can divide that C3 tree seedling group up into encroachers and non-encroachers and get a better understanding of what's going to happen as we go into the future. Thank you.
Thanks, Brad. There's some time for questions, and we've got one from Susie right away.
Thanks, Brad. It just struck me that the encroachers have got more xylem vulnerability to embolism, and they also reduce their stomatal conductance. Do you think there's any connection between those two things?
So that reduction in, we don't know whether they're more vulnerable at elevated CO2. Oh, we do, a little bit more vulnerable. It should be the other way around, Susie. You would have thought that reducing stomatal conductance would make them less vulnerable. So I don't really have an explanation for that, no. I have to think about it a bit.
We've got one in the back from Peter.
Very interesting. What soil was in the pots?
It was a savanna soil, so relatively low nutrients. What we're going to do next is we're really interested in the upregulation phenomenon. So we've got a bigger collection of species, and we're going to grow them in savanna soils and with an addition of nutrients, because we think that maybe species that could upregulate because they nodulated, species that don't nodulate, maybe with a higher nutrient supply, might also be able to upregulate. But we at least want to investigate that.
And here's our last one, Brad.
So Brad, back in the day, Mike Kramer showed that acacia species don't necessarily nodulate, or they do maybe when they're seedlings, but later in life not. You seem to seeing lots of nodulation in your individuals?
No, some species didn't nodulate at all, Steve. So that nodulation thing may well be a Senegalia-Vachelia difference that we're picking up. I didn't have time to kind of highlight all of those differences.
Thank you very much, Brad. Right, folks, we're going to move to Indonesia now, and we've got Johan who's going to be telling us about savanna restoration in Indonesia. Thank you very much. It's a 5-minute talk, and then we will move on.
Okay, thank you. Good afternoon, ladies and gentlemen. It's an honor for me to be here in Savanna International Conference because I'm not a researcher. Yeah. And I have 5 minutes to present my presentation. And thanks to committee, and thanks God for that, because I'm a little bit nervous in front of you. Many great researchers, a lot of great scientists here. Yeah. And my name is Johan Setiawan. I am manager of Baluran National Park, Indonesia. You know Indonesia? Yes. Indonesia not only has Bali, but but also has savanna, like you. Yeah, of course, in our national park. I would like to present my presentation. Savanna restoration and its impact to wildlife in Baluran National Park, Indonesia. I will divide my presentation in three parts. First is introduction, it's about preview of our national park, and the second is challenges, and third is our program approach and the result. Let's go to the first part. Oh, I'm sorry. Baluran National Park is about 29,000 hectares, consists of 25,000 hectares land and 3,000 hectares sea. Our forest stretches from mountain to the sea. A lot of animals in our national park. Baluran National Park, also designated by UNESCO as a biosphere reserve in 2016. Yeah. And let's go to the second part, challenges. Actually, we face many challenges, but today I want to highlight Acacia nilotica. Acacia nilotica is invasive alien species for us, not for you. Yeah? Acacia nilotica was introduced in Indonesia in 1990 as a firebreak and growth massively, booming in 1990s. I'm showing you, this is our national park. 6,500 hectares land of our national park have been invaded by Acacia nilotica, the black one. And large area of savanna ecosystem have been covered by acacia since introduced to Baluran National Park. You can see to the picture, okay, before and after invaded, okay? And the impacts, reducing food source, grass stock for ungulates, drastic change to wildlife trophic cascade. And based on this condition, based on this situation, we've been struggling to reduce massive growth of Acacia nilotica. And our program approach is eradication. We have three schemes of eradication. First, if national park or government program, and the second in partnership, we go with Copenhagen Zoo and local community. The difference between of the three is actually is who is the support, the finance, who the subject is. And these are the methods. Yes, cutting harvest, local community using this method. They cut down the trees and turn wood into charcoal. The barking is government method, and maintenance, of course, grass planting. Okay, you see to the picture. The purple one is area that have been eradicated by government program using the barking method. And the other ones, open area, is local community program. They cut down the trees and turn wood into charcoal to improve the economic. I'm showing you before and after eradication. And the success of eradication is demonstrated by the increasing number of banteng found in camera trap. This is our collaboration research between National Park and Copenhagen Zoo, but not me did it. So don't ask about the method to me. We can see the red box is new data of banteng in 2023. The project's success can be measured by various indicators, and one of them is the reappearance of myna, or maybe starling, or in Indonesia, jalak putih. The myna population tends to increase significantly from 2017 to 2023, start being commonly seen in more areas. You can see to the map, you can see the difference between 2017 and 2023. an egg. And I think this is last slide. These are some study results from several research that have conducted in 2015 and 2021. We have sufficient information to understand the species distribution inside national park. Indicative abundance every species recorded by camera traps in every 1 square kilometer grid system, as well as the prey preference of top predators in Baluran National Park. And thank you very much for your kind attention.
Thanks very much, Johan. We are out of time. We're going to move on to our next talk, which is coming from John. And he's going to tell us a little bit more about the importance of deciduous woodlands in Zambia. Thanks, John.
Right. Yeah, good afternoon, everyone. Thanks for having me. This is my first Savanna Science Conference, and until this exact moment, I was having a great time. So nowadays, I'm a postdoc at the University of Edinburgh, where I study mostly tree carbon dynamics and global change across the tropical Americas, Southern Africa, Northern Australia. But today, I'm going to talk you through one of my PhD chapters from a couple of years ago on the biotic controls of land surface phenology in what I'm calling the dry, tropical, deciduous woodlands of Zambia. So some very zoomed out motivations for this work. There's a need to urgently and accurately quantify the role of terrestrial vegetation in the global carbon cycle. Ultimately, our understanding of these processes will help us to determine the global carbon budget and ultimately the severity of future climate change on us and the vegetation itself. So various process-based modeling studies have predicted that the terrestrial carbon sink is increasing, and they have also suggested that semi-arid dry tropical vegetation is largely responsible for this increasing trend in the terrestrial carbon sink and also the interannual variability of that trend. But there's a lot of uncertainty on these estimates. They're fairly poorly constrained, these models, and so we need to do better. Particularly, we're lacking info on the biotic controls on that carbon cycle process. And one of my main research aims is to try and improve our understanding of the processes by which biodiversity and environment jointly determine ecosystem function. So I'm coming at a lot of this research from older literature from these community scale studies that show there's a positive relationship between tree species diversity and various measures of ecosystem function, whether that's productivity or biomass production or nutrient cycling or what have you. So a bit of background on phenology. So obviously foliage, leaves on plants, they form the primary interface between the atmosphere and terrestrial vegetation. They regulate carbon fluxes, water fluxes, energy fluxes. And land surface phenology is the measurement of that leaf phenology, normally from satellites, normally at an ecosystem level, rather than looking at individual trees. Okay? Have I said everything there? Yeah, so previous studies have shown that there are various fundamental environmental drivers on this land surface phenology, so things like photoperiod, precipitation, temperature, but we still don't have a great idea about what the biotic controls are on those processes, and there's a lot of unexplained spatial variation in land surface phenology. One very interesting feature of land surface phenology measurements across southern Africa is this phenomenon of pre-rain green-up. So a lot of trees in African savannas, woodlands, will produce leaves in advance of seasonal rainfall. And this is a peculiar thing to do because it's actually quite a high-risk strategy for the trees. If the rains don't come, those trees might be forced to abscise their leaves prematurely and impact their growth in future years, or possibly even die. But it might also provide competitive benefits for those trees. So they might be able to beat the grasses if they're smaller. They might be able to beat other trees to the water. So species tend to vary in their phenological behavior. There's a particular group of species, these Datarioid legumes, subfamily of the Fabaceae. These are the dominant Miombo tree species, Brachystegia, Gilbernardia, Isobilinia, that do this pre-rain green-up very well. And there's been a certain amount of ground-based investigation into why they do that and why other tree species don't. So that sort of leads me on to my questions for this study, which were basically, what are these biotic controls on land surface phenology in southern African woodlands? And I came up with 3 hypotheses. So the first, that tree species diversity will promote longer growing seasons and potentially earlier pre-rain green-up, related back to this niche complementarity theory of the biodiversity ecosystem function relationship. The second, that the abundance of these Datarioid species should also promote earlier pre-rain green-up. I was curious to see whether we could actually detect that in land surface phenology data. And thirdly, which I haven't actually discussed very much in the background, is that stands with larger trees that presumably have access to deeper water should be able to green up earlier as well. So to answer these questions, I combined 3 different data sets, which thankfully I didn't have to collect, and I'm eternally grateful to those who did collect them. So I took a ground-based, like a plot data set from the Zambian Ministry of Lands and Natural Resources, the integrated land use assessment data, which consists of many sites across Zambia, each of which have 4 inventory plots where they collected data on tree size, tree species composition, and a few other bits of metadata. I have classified those data there into different vegetation types, just to give you a sense of what kind of species are dominating these woodlands across the region. We only chose woodland plots from that integrated land use assessment, so we're not dealing with grasslands here, we're not dealing with transformed environments, just woodlands. And this data was archived and managed and curated by the COSAW network. If you don't know what COSAW is, they're a network of people and plots across southern Africa. Penny is also going to talk about COSAW a little bit in the next talk. Please come and talk to us about COSAW afterwards if you're interested. The next 2 data sets were a land surface phenology data set derived from MODIS data. So MODIS produce a series of phenological metrics on an annual basis, things like the start of the growing season, the end of the growing season, the peak of the growing season, the length of the growing season. And then I combined those data with the Global Precipitation Mission data, which is sort of the update to the TRMM mission, which gives you precipitation data. And from those I derived various secondary phenological metrics, like the pre-rain green-up as the number of days between the date of green-up from the MODIS data and the onset of the rainfall, the rainy season from the GPM data. And similarly, the senescence lag is what I'm calling it, so how long the trees keep their leaves after the rainy season has ended, as well as the length of the growing season, etc. And the fairly boring bit, I fitted some very simple linear mixed effect models to try and explain what the biotic and abiotic predictors were for each of those phenological metrics. Each of these models, the data point was 1 site during 1 year. So, and then I added site as a random effect to try and account for that pseudo-replication of sites across years. So before I get onto the results of the modeling, I just wanted to give you a sense of like the distribution of those phenological metrics across our sites. So most of our sites experienced some degree of pre-rain green-up. 498 out of 619 sites, not plots, experienced pre-rain green-up in an average year. Season length was highly variable among sites and plots, anywhere between 133 days and 315 days of growing season length. And senescence dates were also highly variable, both within sites and across years, but on average, senescence does tend to occur after the rainy season has ended, which you would expect. So I've blown this up a bit so that hopefully you can actually read. These are standardized coefficient plots for each of those models. So each panel is a model with a different phenological metric as a response, and each of the dots and intervals are standardized effect sizes, where the interval is a bootstrapped 95% confidence interval. So basically, if the interval doesn't overlap 0, we think we found a significant effect of that predictor on that phenological metric. So we do find that species diversity, that's the Shannon diversity index in this case, has a positive effect both on growing season length and also pre-rain green-up. We find that the relative abundance of those ditarioid species also has a similar and independent effect from species diversity on pre-rain green-up and season length. And this isn't to ignore that those diversity controls are still secondary to the effect of temperature and precipitation. Okay, so we're not talking about species diversity overriding or being a bigger effect than precipitation, but it is still a significant, strong effect. I wanted to talk a bit about the senescence metrics in particular. They were fairly poorly predicted by our models, and in reading the literature, there's been relatively little research on drivers of senescence in southern African woodlands, or in the tropics in general. There's a lot more in northern temperate forests where they're talking about autumn leaf drop. But we did find that site had a very big role to play, so we're not measuring something that has a big impact on senescence dates. Maybe it's soil, I don't know. We think that maybe senescence is driven by like short-term oscillations in grass greenness that we're not capturing somehow with our tree data. That's one idea. But we did find that plots with larger trees tended to keep their leaves later, which, although it didn't explain much of the variance in that model, it's interesting and I think demands further investigation. So some implications from this. If you're a biodiversity ecosystem function person, it's quite interesting that we found yet another positive effect of tree species diversity on phenology as a land surface phenology as a measure of ecosystem function. And then independently, we found this keystone species effect. So ditarioid species are driving certain aspects of phenology that other species are not. And so that leads me to two sort of predictions or thoughts on this data. So given that pre-rain green-up is such a risky strategy, and as I think Corley said quite nicely in her talk yesterday, precipitation regimes are becoming less predictable and growing seasons are becoming shorter, at least in Kruger, but I think also across southern Africa. Might we see the decline of these keystone species in the Miombo if they can't adjust their phenological behavior in the face of climate change? And the second, that if we lose biodiversity in these woodlands, will we also experience a reduction in growing season length, for instance? I've got it in my notes. Clara Nutter yesterday said, well, phenology is a really good predictor of forage consumption. So as well as the carbon cycle issues, are we going to start losing wildlife if we lose tree species in these systems? And wrapping up some future work that I've been thinking about as a result of this. So at the individual scale, we need to investigate more what the different phenological behaviors are of different trees. But we can't do that with the data analysis I did. It's very coarse. And also, it's made me think about the limitations of this land surface phenology data in our system, where the land surface phenology is picking up both grasses and trees, and we can't separate them particularly well. So for both of those ideas, I'd point you to the PhenoChange project, which was recently funded, and we'll use remote camera traps to try and investigate individual level leaf phenology and separate the tree and the grass signal. So that's all I have. Thank you very much for listening.
Thanks very much. John, we've got time for one question before you head off. If there is one over there in the middle, are we able to get one, or can I get it to you? I think I can.
Hi, John.
Hi, Diane.
Nice talk. I was wondering, I would have actually thought that trees with, or rather places with larger trees might lose their leaves earlier because it's much harder for large trees to lift up water. So are you saying that maybe all the trees in a particular plot are kind of maybe benefited by some kind of hydraulic lift, or is just the larger trees that contribute to the having leaves for longer?
Right, so my thought was slightly different to yours, is that large trees will generally have a larger below ground hydraulic architecture, and so they'll be more able to get water from deep down, get these groundwater sources. So that's a question about hydraulic lift, right? So I don't know. I think my data is way too coarse to be looking at those like neighborhood effects of large trees on land surface phenology, but yeah, maybe with the phenocams we can.
Thank you very much, John.
Thank you.
Right, so as many of you will know from the WhatsApp group, unfortunately Casey Ryan's talk has been withdrawn, so we're going to move straight on to Penny, who's going to be telling us about the effects of climate change on trees, tree vital rates. Thanks, Penny.
Hi everyone, it's good to be here again. It's been a long break since I've been here. I forgot how intimidating it is actually being up here. So I'm Penny McGraby. I'm the Seesaw project scientist. As John mentioned, Seesaw is a network of people and plots that spans a lot of Africa, and we maintain vegetation plot infrastructure. We did this project in collaboration with the Miombo Network and with the African Forests Forum. So first I'd like to say this has been a team effort, so I'd like to just give some thanks to my collaborators. Many of you will recognize some of the names up there and some of the expertise that they bring to a project like this. So we were interested in looking at the climate sensitivity to the Miombo woodland, and I should hold this thing. So we know that, as been mentioned a couple times before, temperatures are increasing across the region, or predicted to be increasing, and we can actually see that trend when we look at the data. Rainfall variability is also predicted to increase together with elevated carbon dioxide fertilization. And the tropical forest community has gotten a significant head start in understanding the sensitivities of their vegetation to climate change. But this is because they have a long-term network of observational and experimental data. And savannas don't really have access to this kind of really long-term data. And when I say long-term, I mean the Phillips paper is looking at about 30 years worth of data. David Bowman's paper is about 50 years worth of data. So it's significant. And here in savannas, I think because we have these disturbances, we need long timescales as well, because we have some noisy data over time. The reason we need these kinds of long-term data to look at climate change are, firstly, it provides us an indication of what the baseline vegetation stress is. So what is the status of the vegetation before an extreme climate event? It also gives us information about the species level information that gives us an understanding of trait, the level of trait-based understanding that we need to get at veg responses to climate change. Long-term plots also give us an understanding of what the vegetation looks like before and after a climate event. And of course, if we've got this in a plot network, we can do comparisons between sites. Now, in a permanent sample plot where you're tagging an individual stem and then tracking that stem over time, it gets you closer to understanding dynamics on recruitment, woody growth, and stem mortality. Now, I'd like to introduce seesaw here. So as I said, we've got these plots. In terms of once-off plots, we've got about 10,000, but the crown jewels of the network are the 272 permanent sample plots that we have. Unfortunately, at this point, we don't have 30 or 50 years worth of data, but we now have 4 sites where we can look at decadal effects. So we're able to ask the question, what are the decadal effects of climate on African woodland phytodemographics? When we think about the link between climate and phytodemographics, we can hypothesize at the effects on the individual plant level. Now, I'm going to put a strong caveat here. I'm not a physiologist, so I'm a little bit out of my depth, but there are many people here who will correct me. So as a higher temperature means increased evaporation, reduced water availability, and then higher autotrophic respiration rates. And we assume that this would lead to lower net primary productivity, either through some kind of carbon starvation or perhaps water cavitation, and that we would expect to see decreased woody productivity, increased woody stem mortality, and decreased recruitment. Conversely, higher rainfall amounts would lead to more available water and therefore more NPP, and then we would expect to see decreased mortality, increased water productivity, and increased recruitment. But things start to get a little bit more messy when we start to think about the ecosystem and the community scale effects, right? So there are interactions between trees and grasses, and with fire that make things a little bit more messy. So remember I said from an individual plant level, if we increase rainfall, for example, we would expect to see increased productivity. But increased rainfall could also increase grass biomass, which could increase grass-tree competition, create more fire, and then decrease woody productivity. There are some other examples. Sankaran's drought paper has shown that frequent burning could actually remove the grass biomass layer and keep tree densities low, and that would actually boost the remaining trees' productivity. Look, I could go on in this vein for a while, but what I'm trying to say is it's these complexities that give us uncertainties about which directions this is going to go in. So we have these 4 sites. So we've got, this is Republic of Congo up here. We've got a site in Tanzania in Kilwa. This is Nyasa, Zambia just south of Lusaka, and this is some sites in Gorongosa. And as I said, we've got about a decade worth of data, give or take, and we're looking for a minimum of 2 census intervals. So the first thing we did is we took seesaw process stem data, where we're looking at repeat diameter measurements. We're also taking data on whether the stems were alive or top-killed, and whether there were new recruits. We calculated unbiased demographic rates, so this is using the Koyama method. So it accounts for heterogeneous vegetation, uneven age stands, and differing census interval lengths. And then we could have these 3 response variables. So the first is woody productivity, which you could consider as the woody growth of the surviving stems. We've got recruitment rate, which is new stems coming into the 5 centimeter DBH class. And then we've got mortality, which is top kill of stems that we then confirm across censuses. In terms of the climate data, we were using ERA 5 land data, which is a reanalysis product, and we were calculating anomalies relative to a 20-year baseline. So we had temperature anomalies and precipitation anomalies. I'm going to quickly walk you through this. Just quickly, in terms of where we are in climate space, we're spanning a range of sites from about 920 mils to almost 2 meters worth of rainfall, and about 20 degrees in Zambia to 25 degrees in Tanzania. So when we look at the models, this is a linear mixed effects model. We've got woody productivity as a response variable, and here we've got, this is the data for temperature anomalies. So what we can see is that when it gets really hot, we see a reduction in productivity in Nyasa, here in green, and Tanzania, here in blue. Also Zambia, all this very, very messy in Zambia. Maybe if you squint, you can see an increase in productivity here, but I'm not convinced. So when you look at precipitation, again, variable responses across the sites, a decrease, quite pronounced decrease in the Republic of Congo, an increase here in Tanzania, although there's some variation here at the very wet end. But you get the idea. When we look at the overall model, there isn't an effect of temperature and precipitation, but we get these very strong site-level effects. When we look at the mortality data, so this is precipitation, we can see in all bar the Zambian sites, as sites get wetter, mortality rates increase. And it gets, when we look at, okay, so the variation explained on this model is quite high, 68%, but I am going to admit this model is overfitted. It's something I'm still working on, but we'll just move along from that. When we look at the interaction, you can see that in wet years, the mortality rate increases when it's hot. So there is this interaction. Now, do we know what's going on here? Maybe. We've got some colleagues who are working with structural equation models, and they can show when there's high rainfall, the grass biomass increases, and fire-related tree mortality increases, although not rates of other kinds of tree mortality. So we think this is probably a relationship with fire, so it's something we're working on. In terms of recruitment, we don't have a clue. We can explain 0% of this, basically, of this relationship. I think this tells us we're not looking at the processes that affect recruitment at the right scale. We know from the literature that drought effects on recruitment can be on the span of weeks or even months. And when we're averaging extreme climatic anomalies over the period of census intervals, we're washing out those extreme events. So I think this is not the correct method to look at recruitment dynamics. So in terms of take-homes, I think the first thing I'm going to say is these are preliminary models. Precipitation seems to be relatively more important than temperature so far, but we are seeing an interaction between temperature and precipitation. Especially in the woody productivity, we're getting heterogeneous response across the sites, and we can infer that these are from indirect effects, so we're looking at fire and competition over here. These are not the correct approaches to be looking at recruitment data. And finally, I want to leave you with this idea that if we're struggling to see very clear patterns of temperature and precipitation on phytodemographic rates using what are quite high quality, high resolution STEM level data, where does that leave process-based models like DVGMs where they can't account for these kinds of disturbances and don't have this high resolution data, and where they're effectively treating trees like they're in forests? So that's all I have to say, but I'm going to make a quick plug for these three guys. Casey Ryan, Stephen Hancock, and Sean Quigan are advertising for remote sensing postdoc. So if you're interested in trying to unpack woody biomass and structure using radar and then applying that to questions about carbon cycling over the dry tropics, please email Casey Ryan or come and see me. Thank you.
Well done. Penny, you've left plenty of time for questions. So have we got any questions for Penny? Don't all shout at once. Over on that side. Can we get coming? Mike is on the way.
Thanks, Penny. I just wondered if you had any ground stations to check your Error 5 with, because we found for a lot of places in South Africa they don't correlate very well, especially for rainfall is okay, but temperature not.
Yeah, so when we started with this project, we went around in circles for quite a while about which climate data we should be using. Unfortunately, some of the weather stations around these sites there are a lot of gaps in the records. So we elected for something that hopefully had the same bias across the entire region. So yes, we know there's issues, but ground stations weren't really a possibility here. But the other option was using something like remote sensed predictions, like the bioclim data. So yeah.
So you said your models didn't explain differences in recruitment, but could you maybe give us a sense of how much recruitment you picked up and whether it would have been seedling recruitment or what recruitment with that plot data would have represented, given I think you've got a STEM threshold that's already quite a older size.
Yeah. So when we talk about recruitment in permanent sample plots with trees, we're talking about recruitment into that 5 centimeter threshold. So they're already quite old by the time they get in there. So we're picking them up in subsequent censuses. The recruitment data, I mean, I would have to show you the raw data for it, but it is one of our, I'd say, more error-prone sides of our data collection. And I think there can be quite a loss in recruitment between census intervals, so we might not be picking up recruitment pulses and then potentially mortality. The Koyama method is supposed to make some account for that, but yes, I don't know. I can show you the raw data later, Susie. You can look at it.
If there's nobody else, thank you very much, Penny. Right, next we're moving to Tanzanian grasslands or rangelands, and Anna is going to unpack that for us in her 5-minute talk. Thanks, Anna.
Yeah, thank you very much. Thanks for the invitation. It's always a real pleasure to be here, and I always feel so enriched with new ideas and, yeah, go home and start lots of new projects. Anyway, so I'll bring you back to Eastern Africa, to Tanzania. Yeah, this is work in progress. So one of my PhD students, Sabine Baumgartner, is still crunching the data. Stuart Smith, one of my former postdocs, was also part of this team. I'm based at Stockholm University, mostly. So I will talk about particularly traditional rangeland management. And Tanzanian rangelands, East African rangelands are a really special site because they are both hosting a lot of wild herbivores but also domestic herbivores. And as in all rangelands globally, we actually see a big increase in livestock numbers on these rangelands and also in generally higher meat demands. And this is happening on areas, as we've learned, they're very poor in soil fertility. They have to struggle with little rainfall or very variable rainfall. And the plant productivity is highly variable depending on how much rainfall you have available. In Africa, more than 20 million people are dependent on livestock production for their livelihoods. And so this is what we see quite a lot, particularly in Tanzania at the moment. Very large degradation happening, mostly because of overgrazing, because there is still an increasing number of livestock on the ground. And so we were asking the question, well, if we see this happening everywhere, is traditional livestock movement that has happened there for centuries or even millennia, where people moved with their livestock, with their cattle, with their goats, in a transhumance way from one area to the other, is that still a viable option for savannas in this area? And how much resting or recovery is enough for grasses to regrow? So we worked in the northern Tanzanian rangelands in the Simanjiro plains that are dominated by different historical land use, let's say. So we went out with the pastoralists to actually map in a participatory way how they use their rangelands. And we identified rangelands that are only used during the rainy season, so when there's enough rain on the ground. And then there's rangelands that are only used during the dry season, which are these darker greenish areas. And then they also have alalili, which are exclosures, where only very few animals are allowed in, and they're usually sick or weak animals. And so we spread out our study plots along
across these different traditional rangeland uses. We did a lot of measurements, the typical herbaceous layer measurements. We did even combine this with clipping experiments. So we looked into biomass, we looked into grass species or herbaceous species diversity, we looked into soil fertility, grass nutrients, and so on. And we did that over a growing period that was showed very little rainfall and a growing period that had quite huge amounts of rain unexpectedly. And the results are here. And I only have 1 minute to explain these, very complicated graphs, but I thought I'll guide you through it. So if you're looking at these green bars, that shows you that in low rainfall times, in this growing period with very low rainfall, we have very low recovery in general across all the different management sites, and only longer resting and fencing periods actually showed that grass had a positive regrowth here on the y-axis. The blue bars show you that we have generally, it is better to have more resting time, which would mean more than 3 months or a whole season for resting, rather than just coming back after 1 month of resting. Then these purple squares show you these rainy season grazing lands, and they're really interesting for us because they are showing in general very poor recovery. Whatever management you do, the grasses don't really pop up properly again after, even if we have good rainfall. So there's really high attention needed for these rainy season rangelands. And then also an interesting aspect down here, we have the forbs, and the forbs actually pop up really strongly under wet conditions and when we have release from grazing for a while. So it's lots of interesting studies we could do with the forbs, since we've heard that wildlife might eat these forbs even more than we expected in some previous talks, so quite interesting there. So to conclude, traditional movements and exclosures do help lowering the overall grazing pressure, and they do help recovery to a certain extent, but we need long resting periods. We cannot afford making the rotation more intensive than we would want to. And then spatial differences in the recovery potential showed up in these rainy season grazing lands that we have to really take into account when we have future management. And for pastoralists, less desirable forbs popped up. As I mentioned, for pastoralists, less desirable, but maybe a good thing for wildlife on these grazing lands. So my time is up, but I thank you very much for your attention, and thanks to a lot of collaborators and funding.
Thank you, Anna. Thanks for that. There's unfortunately no time for questions, but you can grab Anna this evening or afterwards. Next up is Jesse, and he's going to be telling us a little bit about grass trait responses. Thanks, Jesse.
All right, so I'm going to talk a little bit about a project that I did with a large team of people. The names on the left-hand side are PIs on the project. The names on the right-hand side are the graduate students that were involved. This is a photo of all of us out in the field. This was the rare project I've been involved with all of the faculty and all of the graduate students actually went to all of the field sites and worked together. In the United States, there's a lot of talk about like a mental health crisis amongst graduate students. So for any of you in the room who sometimes suffer like with self-esteem, self-confidence, I don't know if I can do this or whatever, I just suggest you spend a couple months in the field with your supervisors, and like your assessment of your strengths and abilities is going to increase rapidly, and you're going to really feel like you can take over their job probably sooner rather than later. So based on this, one of the driving motivations that we had was kind of thinking about this idea about how do we like think about and describe and characterize grass species. So they're, of course, shorter. They have a somewhat similar anatomical form. And then a lot of times they're overlooked. So we've had 2 days of this conference so far. The word grass has probably been brought up several hundred times. And by my count, there's only been one talk that mentioned a grass species. And you might think to yourself, well, this is just kind of semantic. It's just your feelings are hurt because your like taxon of study basically often sort of gets lumped into one giant heading. But there's functional consequences of this. When we think about making predictions across larger scales, land surface models are typically used based on traits to make forecasts in the future for like ecosystem characteristics. These land surface models were created for temperate forests using temperate forest tree species traits. And at some point in time, some of these ecologists and modelers decided, you know what, we can apply these to grassy species too. All we need to do is collect the grass species trait and then change the model structure such that these grasses are just shorter versions of the trees, and as we all know, grasses are simpler, so we'll just simplify some of the parameters. And I won't specifically tell you how that worked out, but rather I'll give you an analogy. So 10 years from now, at the 31st Savanna Science Networking meeting, none of us will be here because our AI overlords will now be in charge. And they will all zoom in from their respective locations, and one of them will probably propose that they've created a model for giraffes that can predict behavior and physiology of giraffes. And someone in the crowd will say, well, I'm studying humans, and I was wondering if I could use your giraffe model to like sort of predict what the humans are going to do. And what I'm going to do is just shrink the giraffes down and make them more dim-witted. And this model will work really well to predict what Tony Swimmer's going to do. But for those of us like me who are a lot shorter, also equally dim-witted, it's not going to work at all. And so this grass model based on land surface models for forest ecosystems, it just doesn't work. So based on lots of concerns and like suggestions, over the years, the best we've been able to do is to basically get people to incorporate either life stage or photosynthetic pathway into these models. And this has been somewhat effective if you assume that Poaceae basically just requires a simple set of common traits that predict success across large regions. But if you start to think about, can we predict grass dominance, and can we get at convergence and complementarity and bigger ecological strategies, we need a new framework. And that framework is probably going to be going back to one of the very first things, like going back to where you started in this field and thinking about like evolutionary relatedness and phylogeny. So this project is a North American project, but I promise I'll bring it back to all of us in the room here in the beginning. So basically in North America, we have 3 big families of dominant grass species that span the continent. You've got the puids in the north, and then you've got the chlorodoids down in the southwest, and then you have the panacoids kind of in the central east. And these different lineages evolved in different time periods under different evolutionary pressures. The puids are adapted to cold temperatures. They're a cold climate specialist. The chlorodoids are an arid or hot temperature specialist. And the panacoids are somewhere in the middle, this mixed sort of warm environment. And based on these distributions, one of my colleagues, Dan Griffith, has been putting forth this idea that we start to move away from a plant functional type towards more of a lineage functional type. Let's start to incorporate evolution more explicitly into our plant trait relationships. So the first study that we did was on the Konza Prairie, which is just a small 5,000 hectare site in Kansas, but it's pretty species rich. We have over 100 species of grasses in this one small location. And I had a graduate student in my lab, and he measured 75 different species of grasses in this one location, so they're all coexisting in the same site. We did a huge trait-based analysis and started think about how do these relationships vary. The illustrations that are from this paper I have on the right, this is just for the C4. So within Poaceae, we separate based on C3 and C4, and people will often say there's probably some variability in the C3s, but the C4s all act the same. You can see for this snapshot of important traits that are used to drive land surface models, there's a ridiculous amount of variability that exists. Like Steve Higgins mentioned yesterday, I also don't like multivariate analyses. As I'm a plant ecophysiologist, I really can only handle, like, compare this to this. That's sort of like how I enjoy it. But I do like them when they back up the story that you're trying to tell. And so when you look at all of these different species organized by tribes and all the traits we collected, you can see axes of variation within one site that start to get at evolutionary origin, getting at this idea of where these different species and the time periods in which they evolved. So all of this just took place at that one site, but this was the impetus for this project. So we had these 5 large sites distributed from north to south across the continental United States, all coming from different rainfall regimes that span this Puid, Chlorid, Panacoideae sort of gradient. And we had really good representation of species across those 3 major subfamilies. And then for these, we measured a whole bunch of traits. Brad Ripley put up a very similar trait sort of matrix like this. Again, we're ecophysiologists. Instead of coming up with a creative hypothesis, we just measure everything. That's basically how we work. And so we measured all of these. The one thing we haven't done yet, all the pictures on the sidelines, we collected microanatomical data for every one of these species at every one of these sites, so we can get at the plumbing of the leaves and then use those traits to predict what's going to happen as well. So I'll just give you just a couple snapshots so that you can see what I was getting at. The trait du jour right now in the physiological world is Turgor loss point. Turgor loss point is supposed to predict how a plant responds to drought. You can see amongst these subfamilies there's statistical significant variation. You can also see considerable variation amongst those 5 different sites in which we measured. You can see how they are ordered, and this ordering also follows that same patterning of ordering of this variable, follows that same ordering and patterns we see with evolutionary origin, where the more mesic panacoids have a higher Turgor loss point, a lower drought tolerance, compared to the arid land specialists of the Chloridoideae. So you're seeing these things sort of play out in real time. This one's photosynthesis. Photosynthesis, this should be the main variable that separates C3 from C4 species. And you'll notice that there's a lot more variability within sites than there is across these lineages. The exclusively C3 lineage, the Puidae, is actually lower, but you can see it's not nearly as much lower as you would think. This next slide is my favorite because there's one variable that all trait nerds, regardless of like what field you're in, measures, and for a long period of time I've complained about this, and it's SLA. And the one variable we measured, and SLA functionally means nothing, and it didn't have any variability at all. I felt like a career of crapping on SLA was like finally like validated. So you can start to do these bivariate relationships and start to think about how these vary through time as well. And this one I think is quite nice. This one we're comparing photosynthesis measured with gas exchange with electron transport rate measured with chlorophyll fluorescence. So 2 different techniques. In the panacoids you can see strong relationships, weaker relationships for the 2, and then no relationship at all for the one of them. This gets at this idea, if these were all grouped together under this one broad envelope as grasses, it probably you wouldn't tease any of this out. You can also look at the classic relationships of thinking about like how 13C, which is often used in C3 species as an indicator of water use efficiency, how that might vary with this common trait of thinking about drought tolerance, so water use efficiency and drought tolerance. And historically, this is only done in C3 species, and C4 species are just presumed to have no variability whatsoever. You can see that we didn't see a relationship for the C3 species, and then the C4 species, we see a tight relationship between 13C. On this C3 one, if you remove the panacoids and just look at puids, you can start to see those nice trends. So again, sub-responses vary by subfamily, and they're sort of playing out based on what evolutionary lineages would suggest. Similar to our site-based one, if you look at a multivariate analysis, you can see these axes based on resource availability and lineage playing out in terms of how traits are organizing basically across evolutionary space. So hopefully with this, I've given you a little bit of perspective that to hopefully in some capacity is encouraging you to move away from a plant functional type framework towards one that's more based on a lineage functional type. and that this idea of C3, C4 is just an insufficient way to do this. However, most of you aren't plant nerds, so Ben and Sally and a few other people in the room are probably really excited because they are big plant nerds, and the rest of you are like, whoopee, who cares? And so the one thing, because to get beyond academic curiosity, you have to think about how do we scale this and make this more relevant. So how do we take what we've done so far and we increase that spatial representation, how we improve actual models, and then we start to gain insight into places that aren't typically measured for a variety of reasons. They're either inaccessible, they're unsafe, or there's a lot of reasons why. So to do this, what people have been proposing for a while is to use hyperspectral data, and you can measure hyperspectral data at a plot scale. Hyperspectral data is this idea that leaf reflectance across the broad band of light should vary based on how a leaf is built. So the patterning of cells and the types of materials that are in cells, and maybe you can use these hyperspectral profiles and link them to phylogenetic relationships, and then you can start to move up from a plot scale to a landscape scale and start to make predictions. So for every one of these locations that we measured these traits, we actually went out and did full hyperspectral profiles on every species at every site, and then we did plots, 100-meter plots at every location replicated to get at mixed assemblages as well. And one of the things that we found is as you start to look across the broad spectrum, you can pull out phylogenetic lineages in these major subfamilies of grasses. We just published this in the last month in JGR Biogeochemistry. And the nice thing is if you use basically a linear discriminant analysis, it was almost a 100% predictability about thinking about how these different lineages parse out. So this means that if you're using this broadband hyperspectral data, you can pull out the lineages of the grasses that are in this picture. This is a big improvement considering that historically, remote sensing data has a really hard time differentiating not only grasses from forbs, but all plants of similar heights. So if we can start to use these data products to do this, there's huge avenues of possibility. The last thing that we've done is used all of this hyperspectral data to start to see can we actually predict different traits. So the last thing that I'll point out is some work that Stephanie Powell, who's at Berkeley, is leading. And Steph has come up with basically using our hyperspectral data to compare predicted trait variables with all of those ground truth measured trait variables. And for a lot of the variables that are important for driving land surface models, we have pretty good relationships. And so if you're trying to think about the significance of this going forward, the next few years are going to be pretty exciting because a lot of space-based observational patterns, observational sensors are being launched right now. So we're not only going to have vast improvement in terms of the spatial resolution on the ground, we're going to have this broad spectral inference to start to be teasing out exactly what species and potentially what traits exist in those species for huge portions of Africa. So with that, take any questions.
Thanks very much, Jesse. We've got time for one question. Dominating here, Susie. It's all right.
Okay, that was absolutely fascinating. I just wanted to know, is there any relationship between those C4 lineages and photosynthetic subtype? Brad knows the acronyms better than me, but you know what I'm talking about. NADP,
Sure.
Yeah.
Yeah, so C4 species have different decarboxylation pathways, so that's what we would call C4 subtype. It's how CO2 gets re-released around the main carboxylating enzyme. And a lot of times those are conserved in tribes, and so there's not many in which they would have multiple subtypes. So that ends up being a subset of this idea of tribe, basically. So that same information gets conserved in tribe, but it allows you to draw inference into how evolution structured a lot of these processes. Does that make sense? Absolutely. So for things like bundle sheath cell leakiness, that's very much a C4 subtype phenomenon, and it explains why the Chloroidoideae, have that relationship with turgor loss point. It's based on bundle sheath leakiness, which is a function of their C4 subtype.
Thank you very much, Jesse. Give him a round of applause. Right, we've come to that important part, the last talk of the session and of the day, and the bravest person in the room is giving it. Tsitsi is going to be talking about marula leaf traits. Thank you very much.
Pressure after a fantastic talk like that. Okay, so like he said, my name is Tsitsi Maponga, and I'm here to present on how leaf traits of the host trees, Sclerocarya berea, vary across a rainfall gradient and level of mistletoe infection. So as we know, large trees are keystone structures in African semi-arid savannas, and some of these large trees are now surpassing their physiological thresholds. So this is due to erratic rainfall, rising global temperatures, fire, soil resources, human activity, and so forth. But mistletoes are now making these large trees more susceptible to physiological stresses. But what are mistletoes? So mistletoes are hemiparasitic plants that form an intimate connection with their hosts, and this enables the mistletoes to efficiently take up the water and nutrients from the host. However, this relationship between the mistletoe and the host can generate physiological, morphological, and phenological stresses. So we aim to investigate whether rainfall and severity of mistletoe infection influence the leaf traits of the marula hosts. So we sampled 3 villages, that was Seville, Dumfries, and Xanthia. So each had a mean annual rainfall of 590 millimeters, 655 millimeters, and 695 millimeters respectively. So all these 3 villages are located in Bushbuckridge in the Mpumalanga province in South Africa. We had 3 infection categories. So that was uninfected, low infected, and high mistletoe infected trees. So due to the limited amount of time for this presentation, we only show the results for the leaf traits that are directly linked to nutrient use and acquisition. Of course, we have specific leaf area, leaf dry matter content, and leaf mass per area. So we defined our acquisitive resource users as having higher specific leaf area, lower leaf dry matter content, and leaf area. All these are linked to higher photosynthetic rates and plant productivity. We also defined our conservative resource users as having higher leaf dry matter content and leaf mass per area, but they had smaller specific leaf areas, and they also have high resource use efficiency, but they would rather invest more in structural attributes rather than their growth. So our results show that all marula leaf traits, the 4 leaf traits that I mentioned, were influenced by rainfall. However, only marula leaf area and specific leaf area were influenced by mistletoe infection intensity. And the interaction of rainfall and mistletoe infection had significant effects on all the marula leaf traits. So when it came to rainfall-specific effects, marulas were resource conservative at high rainfall, and this was possibly due to the depletion of soil nutrients from the leaching from the high soil erosion that we observed at this site. But you can see that at the intermediate rainfall site, marulas were more resource acquisitive, and this could be due to the availability of nutrients leading to high productivity and photosynthetic rates. And when it came to specific mistletoe infection effects, marulas were more acquisitive at high mistletoe infection, possibly to increase their competitive capabilities. And indeed, in one of our studies, we saw that there's high nutrient and water availability below high mistletoe infected trees to support this kind of behavior. And in contrast, low mistletoe infected trees were rather conservative, suggesting that they could be responding to nutrient and water stress, and possibly due to the inception of the mistletoe infection. So we can say at inception, mistletoes are more harmful as they are directly acquiring like the dissolved carbon from the host. And in terms of the interaction of rainfall and mistletoe infection, highly infected marulas in the intermediate rainfall site, obviously from what we've seen, were mostly acquisitive, probably due to resource availability, as we've mentioned, and also to increase their competitive capabilities. Then low infected hosts at the high rainfall site were mostly conservative due to low resource availability and as a response to mistletoe inception. So we can see that variations are not always clear-cut. Other factors such as soil nutrients that were not directly measured here could be causing these differences. However, the broad range of leaf trait expression of the host could ensure that marula survival in cases of stochastic events, could ensure the survival of marulas in cases of stochastic events. So we can we can safely assume that the heterogeneous rainfall and mistletoe infection in bushbuck ridge can support greater plasticity of the host leaf traits, thus the persistence of the marula population. So we'd like to thank the NRF and Zion for the funding to present at this network meeting. We'd also like to thank Mighty Man Mashele and Frank Nyati for assisting with the data collection. But please follow me on Twitter or X. I'm Mistletoe Twist. I'm less nervous on Twitter, so yeah, I think we can even discuss outside after this whole presentation. Thank you very much.
Sorry about that little slip there, guys. So who has got questions for Tsitsi? She left us with plenty of time. I know people are eager to break out of the air conditioning and go and brave the heat outside. Tsitsi, did you need to standardize the size of the trees that you used at any of your sites, or did it not matter?
No, it mattered. So all the trees that we measured were of equal size. Statistically.
Perfect.
Yeah, but so I'll be presenting again. There's a seminar on the 4th of April. You can search the Zion website. We're going to present more on the results from my PhD and what we found now. So, yeah, since you don't have questions.
Thank you very much, Tsitsi. Thanks a lot. Just two final announcements before I release you to the beers.